Hi, I wanted to welcome you to the Congressional Biomedical Research Caucus. This is our 20th year of having these caucuses. I'm Joan Goldberg, Executive Director of the American Society for Cell Biology, one of the members of the Coalition for the Life Sciences, and the CLS works with the caucus to put these briefings on science available for Congress and congressional staff to help you in policy decisions. Today I'm very pleased to introduce Dr. David Botstein. As both a visionary thinker and what we call a bench scientist, somebody working in the lab, he's contributed in far-reaching ways to genetics, genomics, and science education over nearly 40 years, and I say nearly because I didn't exactly look at the dates there. He has played a fundamental role in basic discoveries about bacteria and yeast. He helped lay the foundation with colleagues for the Human Genome Project, the full mapping of all the DNA we possess, which is approximately 35,000 genes. Dr. Botstein's major contributions also include the classification and study of human tumors using microarray technology he developed with Patrick O'Brown. He earned his AB degree from Harvard, his PhD from the University of Michigan, and began his academic career at MIT. After serving as Vice President of Science at Genentech, which has been in the news lately, from 1987 to 1990, he chaired the Department of Genetics at the Stanford University School of Medicine. In 2003, he assumed the position of director of the Lewis-Sigler Institute of Integrative Genomics and the Anthony Evnin Professor of Genomics at Princeton. He also leads the Center for Excellence in Complex Biomedical Systems Research, also at Princeton. That center provides infrastructure for research and teaching programs where biology, physical sciences, and more quantitative sciences meet, which is an increasingly exciting place to be, both for people learning science and people doing science. I'm pleased to welcome Dr. Botstein, who will address how the genome and the computer have reshaped our view of cancer. Thank you very much.
I need to see this, so can you hear me all right? People don't usually have trouble hearing me, so. Thank you very much for the opportunity to talk to you, and I'm going to give you a very brief tour of what the title actually, I didn't write the title. Mike Bishop wrote who organizes these things, wrote the title. And usually when people write titles for you, you say, oh, God, I don't want to do that. But this was actually perfect because it is exactly what I think is a reasonable thing for me as opposed to some other person to talk about. And so I like it a lot. And the thing I'm going to emphasize, and sort of the theme that I would like you to be thinking about, is the issue of how does computation and mathematical analysis and this kind of activity, how important it has become in doing even the most basic ordinary science, and in turn, how important the most simple ideas of science are when you're actually applying the computer. If you do it blindly, then you're not going to get anything helpful out. And so that leads to the need to educate our scientists differently than we did in the last century. And so that's where we're going. So I'm going to start with the genome. You've probably seen slides of this kind many times. Each of these things is a organism that is whose sequence is now fully known, thanks to the Human Genome Project. And this is the number of genes that encode proteins in all of those organisms. And as you see, yeast, which is beer, bread, and wine, is very small, but still has a lot of genes. And a human and mouse have very large genomes, but they don't have that many more genes. And that, of course, is one of the sort of results that got in the paper. Now, the thing that didn't get in the paper, for various reasons, which is a major theme that I'm going to pursue here, is that the basic cellular functions, what each of these genes and the proteins that they make do, is conserved over evolutionary distance. And what that means is that a gene in yeast and a gene in the human are not only conserved in their structure, but they're also conserved in their function. And I'm going to tell you what that means and why it's important. So in order to, this is how business gets done these days. We know about genes, we know about proteins, and we know about what they do. And we want to try and connect all these things together. And in order to connect all these things together, at the end of the day, no matter how much computation you do, you have to do an experiment. Somebody has to be mixing up test tubes or the equivalent in order to know that this particular protein does this particular function. However, because we have molecular biology and sequencing, we can make connections between gene and protein in a very simple way, the genetic code and things of that sort. And we can also, in model organisms, we can remove genes, or in diseases, genes are removed for us, and from that we can infer function. But somebody has to do that kind of work. Given that that kind of work is there, we got a huge favor from nature. And that huge favor is sort of encapsulated in this slide. Basically, there's a list of proteins. It's not important what they are. This says to what extent the amino acid sequence, proteins are polymers of amino acids, is identical between a yeast and a mammal. The mammals to this level of resolution are all the same. And as you see, that varies a lot, from about 25% up to 96%. This protein was actually named for this feature. But actin, is what moves your muscles. Yeast don't have any muscles, but they have an actin that's 89% the same as that of yeast. And this table of conservation is now extremely well worked out because of the Genome Project. And the sequences of the many organisms. But even more important is an experiment that you can do, because in yeast, there is no yeast rights group. There's no ethical issues around manipulating the genomes of yeasts. And as a reason, for that reason, it is now available. You can get every gene of yeast removed, just that gene. And so then what you can ask the question, well, what does that do to the yeast? Well, most of the time it means the yeast is dead. What you can do now is you can take the mammalian sequence and put it there in place of the gene and ask what happens. And wherever I've written yes, the answer is the yeast come back to life. So to put this in very concrete terms, let's suppose that out front here, if you have the parking sticker, you have your modern car, let's say some kind, the latest Buick or something, and you have a Model T Ford, okay, let's say, and the Model T Ford won't run because a part has fallen out of it. What are the odds that you can reach into the modern Buick, recover a part, slap it into the Ford, and drive away? Well, there's no chance of that. And the evolution of cars is only about 100 years. They started making cars at the beginning of the last century. So these organisms are separated by 1 billion years, or give or take, of evolutionary time, and the parts still work together. That's pretty remarkable. Well, we learned that in detail from the Genome Project. So I give a much longer talk about what the fruits of the genome are, and I've made these bullet points. One, which I'm going to talk more about, is a quantitative understanding of evolution from sequence comparison. Another is this grand unification of biology, which I've already told you about. Then I'm going to talk about how you can use DNA sequence differences, little differences, not only to do forensics, which I'm not going to bother you with. You all watch Law and Order and stuff like that, and every plot is the same. It all ends up in DNA comparison. But I am going to talk about how you can find disease genes. And then those of you who came here for cancer, it won't be so long from now, will be happy, because then we're going to talk about cancer and what all this means for cancer. Now, the first thing you have to recognize is that this sequence comparison business, this table, is not so easy to produce. wait, I got ahead of myself. The evolution piece, this is a drawing that Charles Darwin, whose 200th anniversary, right, made right after he came off the Beagle, within a year or two. Okay? And this is actually what the Genome Project say that is the thing. It's remarkable, the intuitive insight, 1837, and this is the way it really looks. Not so different. So he really did have the right idea. And I want you to notice that the animals and the fungi, okay, are the most similar in this tree, okay, and the bacteria and so on are way out here somewhere. And nevertheless, we can trace individual genes through this entire tree, and when we do that, we learn something really useful. Now, the point I was going to make before I got interrupted by Darwin was that, of course, lining up these, this is a particular gene, it's called mut-S in bacteria, and this gene is present in every organism on Earth in some form or other. In many cases, there are more than one copy. And in order to get this information, what you have to do is you have to align the sequences. And this alignment is both mathematically and computationally not trivial. And not a small amount of computer, you ask why the NIH, on whom we all depend, okay, has to spend so much money on computers. This is just the beginning of why that has to be the case. And why we have to teach our students computer science is because this problem, although it's solved in a rudimentary way, is not solved to the extent that we really need it to be solved to be fully useful. Anyway, what you can see is, and you can't see, but I'm going to read to you, that this is a yeast one, this is a mouse one. And these are arranged in order of similarity. Yeast, mouse, human, yeast, bacteria, another yeast, and so on. And you can make this into a tree, just like Darwin's tree. And when you do that, you find that the mut-S's, in fact, have proliferated through evolution, and that higher organisms have many different mut-S's. And these, when you look in the lab, you see that they actually have different functions, slightly different functions. They all are involved in repairing DNA damage, but they do it in different ways and under different circumstances. So this is the basic mechanism of evolution. Duplication, bacteria have one, the higher organisms have many, and divergence of functions so that you can get a much better solution to the DNA repair problem. Okay, now, how do we get information out of the human genome sequence? Well, one thing is by finding disease genes, and I'm going to talk about that. Another way is by this comparative genomics, and I've basically said everything I'm going to say about that. Then there's patterns of gene expression, which is where the big impact has been so far on cancer. And then there's this other stuff, which I'm going to skip. Okay. The thing about DNA that is so remarkable, that makes all of this stuff possible, is that it has a chemical ability to recognize itself, its complement. So there's a rule, C's go with G's and A's goes with T's and G's and so on, and that actually in a test tube in chemistry, you can get this reaction to going from single strand to double strands. This is the famous double helix. You can get this to go if and only if the sequences are the same, are the true complements of each other. Okay? And if there's a mistake here, then this reaction goes badly compared to if there's no mistake. So this allows us to recognize in the test tube as well as in the computer the similarity between two genes. So when we do that, and this is using radioactive probes, it's not the way we do this involves hybridization, but the details are unimportant. You can actually follow a gene in a family. So Ms. 4620 over here, there's a whole bunch of random people from Utah, and you can see they each have two bands, and they're in different places. And here, Ms. 4620 over here, 4620 has a band here and a band there. Her husband, 4621, has these two bands, and they have 3 children, 4622, who has the lower band from each of his parents, and these 2 ladies, young people, have the upper band from mom and the upper band from pop. And you see they're identical in that regard. Now, if that marker is right next to the cystic fibrosis gene, and the parents were carriers, then if this girl had it, then this girl will have it too. And that's the principle of DNA diagnosis. Since this was introduced in 1980, you can see an exponential increase in the number of genes that have been found in this way. And today, this statistic is true. I heard yesterday at a meeting at NIH that the big number, has become now 7,000, and so there's more to do. Okay. Now, what happens when you find a disease gene? What happens when you find a disease gene? You learn something about why it causes the disease. So in the case of Huntington's disease, a whole class of diseases was found which are mismatch repair defects. You can find really surprising things, that in the brain, okay, that the problem that causes many brain diseases is really about having oxygen running around loose. No one had a clue that this was even an issue before this gene was found. This was not such a surprise, that cancer has something to do with the cell cycle, and then that the cancer can be caused by loss of function as well as by gain of function was discovered from identifying that gene. So, I'm going to talk for the rest of this time about examples of genes that somehow cause cancer. And one of the genes, a gene that was mapped in families that have predisposition to colon cancer, turns out to be these members of the MSH2 family, which is the family I told you about that had duplicated and diverged from the most ancient bacteria. These two are related to each other because they are oncogenes that were discovered in a different way, but originally with the so-called Philadelphia chromosome, where there's a translocation between this gene and an immunoglobulin gene, so that gene is inappropriately expressed. And finally, this gene it was found in families that are predisposed to breast cancer, and this gene, quite nearby, is the target of one of the first drugs that actually does some real good for cancer patients because it actually addresses the cause of the disease. Okay, I'm going to skip that. In September of '93, this paper appeared, which said that there was a mismatch repair defect in tumors, colon tumors. And this was an interesting paper. However, we'd already entered the genomic era, and there was a scientist in North Carolina who read that paper and said, I could find mutants in yeast that do the same thing, that destabilize the tracts, right? So he did, and he found the genes that are called PMS1, MLH1, and MSH2, okay? And then that was in September. And by December, the gene had been found by the method I outlined, and it turned out to be MSH2. Turned out to be the same gene, and that in fact, this whole thing got done in less than a year because the basic science was understood at each step. It turns out that 90% of all the familial HNPCC families have mutations in either one or the other of these homologs of MutS. Finally, I'm going to tell you about another use of hybridization to classify tumors. And the idea here is very straightforward. what we have here is an array of, on a glass slide in this case, it doesn't have to be glass, in which we have every gene represented of the human. And what we do is we ask the question of a cell, are you expressing each and every one of these genes? And whether or not they're expressing it is reflected by the color that comes out when you take a picture of the final result. And our brains are not capable of looking at a table of numbers, which is a few hundred across and 22,000 down, and making any sense of it. In fact, you can't get it into Excel. Microsoft won't let you make such a long sheet. Okay, so we have to do something else, partly to make Bill Gates happy, but partly to make the rest of us happy so that we can understand the result. And so what we sought to do was to change the numbers into a pattern. And what we did was for each row in the table, which represents the genes, we figured out the mean, or the median, and then we characterized each cell by whether it was above or below the median. Below the median is more green, above the median is more red. And then we have all our samples across the top, and now if you line these up, you can see, without doing any numbers, that whatever this experiment was about, it went from low to high for all of these genes. Okay? That's the idea. All right. So we seem to have, okay. So there are methods that are well known, and in fact, it's very similar to the method that was that we used for figuring out the trees for evolution of MSH2. We can then take these patterns of gene expression that we see, and we can figure out how to cluster them. And these two are more similar than that one, and so on. And to give you an idea of how well these things work, imagine that this famous picture in the Louvre, which we made black and white to simplify the problem, was a series of stripes that are like these genes. And now we randomize them. Okay? And now you can't tell anything. You can't see the Madonna, you can't see John the Baptist, you can't see Jesus, you can't see the tree, you can't see the sky, you see nothing. Right? Now you apply the algorithm, first step, second step, and while it's not perfect, you get a pretty good idea, and if you've ever seen this particular Raffaello, you would recognize it immediately, maybe even from the first step. So that's basically what we're ordering the data, whatever, we're trying to extract whatever order there is in the data. This is computationally, you can't do this by hand. There's no way. This is a computational method. And when you do this for human tissues, you get these pictures, which now that you understand that in this case yellow is up and blue is down, you can see that each of these tissues across the top is different. You can see that the pattern is different, the patterns of yellows and blues, but some of them are more similar to each other than others because I've clustered in both dimensions. So now let's do this for tumors, and you get another really nice result. The really nice result is that all the breast tumors are more similar to each other than they are to anything else, which means that they remember that they came from the breast, all of them. And the ovarian tumors are different, and the leukemias are different. Okay, so that's nice. But you can do better than that because if you look at higher resolution at the breast tumors, you can see that there are different subtypes, at least by this classification, of breast tumors. There are the dark blue ones, the light blue ones, the magenta ones, and the red ones. And so there's a lot of argument about what does this mean, and very important in science is replication. And so this was a bunch of Norwegian and Californians, and so we looked at another data set which were Dutch. And when we reanalyzed the Dutch data, what we found was that by and large the red ones stayed together, the light blue ones pretty much stayed together, and the dark blue ones pretty much stayed together. Very good for this kind of statistical stuff. Again, you can imagine there's a lot of computation that goes into doing that comparison, and some statistics, I should say. So we were very confident that this classification is meaningful. And so now you can ask, well, does this have any clinical meaning? And the answer is, well, yes. In the Dutch data set and in the Norway set, the dark blue ones die much less rapidly than the light blue ones, who in turn die less rapidly than the magenta ones or the dark red ones. Okay? Remember the magenta ones because they are ones who have HER2 amplified, and there's a drug for that, and I'm going to tell you about that. Okay, so now we can make a basic science question out of this. We can say, if this classification is true, and that these tumors are really fundamentally different from each other, then if I have a mutation that predisposes to breast cancer, I expect that they should be of the same type and not of 4 different types. And so you can do that in the computer. I can just take the data I already have, add one extra fact, who inherited the BRCA1 gene, and we then relook. And we did that with the Dutch data set because Van de Veer had this information. And what you see is that the BRCA1 carriers are all the dark red type. So this is biologically, genetically, as well as clinically significant. This is, at this point, just an anecdote, and in fact, this is misleading in practice. You can't believe anecdotes of 2. This is way more than 2. This has high statistical significance. This has none. Okay? All right. So this is obviously a good thing to be able to do. You can distinguish tumors, and that means you get better diagnosis, you get earlier detection, you get new targets, because each of those things is a gene or a protein. And one of those was that HER2, the magenta class, and another is this ABL or KIT, and that for breast cancer, for different kinds of cancer, and then it's useful in clinical trials. Here is the data from the FDA originally that was submitted to the FDA for adjuvant treatment with the drug against the magenta gene. And I have to disclose, in fact it was already disclosed, that I used to work for Genentech. I had something to do with this way back. But the reality is that what used to be one of the worst tumors has now become no worse than the light blue tumors, okay, because of this treatment. And in the same way, the chronic myelogenous leukemia directed against the ABL, they have even better results in terms of survival. This is survival. So, to summarize, we have many ways of using the genome in the computer to find genes. When we find genes, because of the evolutionary conservation, we can apply all kinds of basic science information that we learned in the model systems, like that this is a repair gene, right, to understand why it's causing cancer. We can look at similarities in proteins. These 2 proteins are related to each other in the same way that MSH2 is related to MLH1. And so we can use this information, and actually in the clinic, when Gleevec came out, okay, somebody thought, you know, KIT is just like ABL. Maybe it'll work against a GI stromal tumor. And it works almost as well against GI stromal tumors as it does against these leukemias. And finally, in breast cancer, you can see that we have a drug that works. We also have a way of using the inherited information to know that our classification of different types of tumors actually has biological meaning. And so for the future, of course, all this genome and computer raises some issues that I know people are interested in, the personal genome, and the very important business that this is such a probabilistic business that the doctors and the patients need to have some reasonable intuition about how to think about probabilities. And then, of course, you have to think that in some cases, the information that you carry a gene is of no immediate use. And so the question is, who do you want do you really want that information? And then in other cases, like in the case of the MSH2, it makes a huge difference because most of us don't enjoy colonoscopies. Those of us who have an MSH2 defect understand that we better go pretty frequently because the probability that we're going to get one of these is not small anymore, right? And in the case of HER2, of course, it makes the difference between having a diagnosis of breast cancer, that is, for which there's nothing to be done, as opposed to one for which there's a lot to be done. And with that, I'm done. Thank you. Was I on time? Okay. Hello, sir. It's my congressman.
If I may come forward?
You may indeed. This is your house, right? Your other house.
And please excuse me, we're in the middle of a floor debate, and we had votes, and I had wanted to be here to introduce to you Dr. Botstein, and I hope whoever introduced him did justice to his really distinguished career. The Genome Project would not have been possible without his early work. His clinically relevant work, some of which you saw here today, is probably already affecting, I can say without fear of contradiction, is already affecting people's lives positively. And it hits home right here this week because many of us know our colleague, Representative Wasserman Schultz, actually has over the last year faced decisions about genes and breast cancer and uterine cancer and other such things. What I wanted to talk about that other people might not have talked about in introducing David Botstein is how outstanding he is, perhaps unique, in his ability to combine disciplines. You got some sense of that. The statistics, the mapping, the graphical, and so forth. But to combine it even further into working with policy people as well as the science people. And this is what is of particular interest to me. He is the creator of a new concept of education in college. In fact, President Shirley Tillman recruited him to Princeton so that he could carry out this new educational idea. And it is to combine biological, chemical, physical, mathematical sciences, computer sciences in introductory courses all at once. And the students are responding very well. It may, I think it's only at Princeton so far, although he has published and people are probably picking up on the idea and its success.
Yeah, and it's not, to be fair, there are some small colleges that have been doing this because they have only 3 science teachers, and that's how they do it, okay? And in fact, they have historically had a lot of success. It was part of the reason for thinking it might be a good idea.
But it gets at the idea that this caucus, Biomedical Research Caucus, is built around. It is recognizing that it is the days of a single scientist in a narrow discipline are past. And NIH has generally kept up with this. NSF has generally kept up with it. But it is still not, widely understood in universities around the country that what you need and what you want and what students respond to is this curriculum that is as multifaceted as today's research is. And David Botstein has pioneered this to great effect. So this may have more benefit to society in coming years, even than the research that he's done so far, because there are now hundreds, I guess, not yet thousands.
Not yet hundreds.
Not yet hundreds.
Dozens.
Okay. Okay. There are
hundreds to come.
Hundreds to come of these trained undergraduates now loose and affecting the nature of research. So I did want to say something about that. Now, a few more words about the biomedical caucus. We exist not just to have bag lunches and interesting presentations. We exist to keep up here in the Congress the support that's necessary for the research that will save lives and relieve suffering in the short term and the long term. And some of that research is done at NIH, and some of the research is done even at the Department of Energy, even at certainly at the National Science Foundation, and through other government sponsors, and of course in industry as well. And but when I say the support for these things, I don't mean just money. It is particularly nice right now to recount how the money is going. In the Recovery and Reinvestment Act, NIH got an additional $10 billion, new money for research and for laboratory research and laboratories. $3 billion to NSF, $500 million to NIST, and quite a few other billions of dollars. Altogether, about $22 billion in the Recovery and Reinvestment Act. And in the budget that the President has sent over, science continues to do very well. And we certainly want to work to sustain that. But also, we need to make the point with your offices, with my colleagues, that the environment for productive research requires more than money. The President has said in his inaugural address, we will restore science to its rightful place. That is a sentence that's fraught with meaning, lots of different meanings, all of them good. And so not only do we need to sustain the dollars for the research, but we have to make sure that we don't let narrow ideologies get in the way, that we don't let government or industrial corporate censorship get in the way. And we have to make sure that we are directing and attracting the kinds of students that we need, and giving them the kinds of courses, the curricula that will allow them to do the innovative research that somehow David Botstein managed to do without having gone through his innovative curriculum. So now let me turn it over to your questions.
Thank you.
Thank you.
Thank you very much. Yes, ma'am.
You mentioned colonoscopies
Yes.
and that they're no fun, and they're
Yeah.
also not cheap.
Yeah.
And I know Congresswoman, is it Wasserman, the woman who's just had all the cancer surgery?
Yeah.
She was saying that her case has demonstrated that annual mammograms in some cases should be standard of care way before the age of 40.
Yes.
And that, of course, is not cheap. So my question to you is how soon will this kind of work that you're doing make it possible to fund those sorts of tests as often as necessary for the people who need them, and fund them much less often for the people who don't?
Actually,
Saving federal dollars.
Actually, it's a better question for Congressman Holt than for me. I'm just worker bee at this point. I do think that the way in which our science impacts is that for a very large, so the way it's being used clinically already is that, for example, the dark blue women don't have to have chemotherapy. And the statistics say the chemotherapy does them little or no good. And so what's happening is that they're not doing chemotherapy, they're having tests, and then they're not doing chemotherapy, and that ultimately saves money. So I think that what our science can do is it can point to the people who are at highest risk and basically rank them from highest risk to lowest risk, and then have the appropriate diagnostic, more expensive diagnostic things parceled out in accordance with the risk in such a way that a woman who has a very low probability of getting breast cancer doesn't get mammographed and to the point where actually the cancer is being induced by the radiation more frequently than any is being detected, and the women who are at high risk get the mammograms as soon as they should. That's, I think, how the science will impact. The business about where the money is going to come from is, as I say, above my pay grade, although I do think that I'm very optimistic that a real, serious, universal health care system will make it much easier to provide these services to people as they're needed. That's...
But I guess when I said how soon, how soon
I have
would
no
this
idea.
be? Okay.
Yes.
I met with a man yesterday who's the director of business development at a very large firm. Everybody would recognize his name. It makes a variety of biomedical products. And he is in their diagnostic. He said they're very much limited in what they make because they are not going to invest millions of dollars to come up with an assay, for example, for a gene, a protein, if there's going to be no market for it. And there's going to be no market for it because right now the insurance companies won't reimburse for these tests because there is no practical endpoint yet for them.
Well,
and so he said his firm is spending all of its money, this is a major big firm, spending all of its time making improved assays for the proteins and markers that we already know.
Okay, so I have two answers for you. One is that's why the startups have regularly been killing the big old firms, okay? Because the startups don't believe that, okay? And in fact, there is such a startup, several such startups who are doing the breast cancer thing, right? And your friend won't do it. Of course he won't. That's one answer. The other answer is that I don't think that there should be diagnostic tests loosed on the population until you have a clear idea. That was the point of my last slide. Until you have a clear idea of how it's going to be used. There's no point in getting information that no one is going to use except for research. And for research, you don't need commercialization. Yes?
You did allude to who might play a larger role in some of those questions about probability when you have statistical geneticists as a question mark for that point.
Yeah.
Right now, for people who already have diseases and have different types of breast cancer, there are an array of tests that can give you information, but you get information in terms, as you said, of probability and risk that most doctors and patients can't understand.
Well, don't understand, yeah. It's an issue that we all face, and it's partly an issue for the universities and for the medical schools, and it's partly an issue for education as a whole. We are not very good at teaching probability and statistics altogether. Otherwise, the casinos would not be so full. It's a problem, right?
I was wondering if some of the more computationally trained students coming out of Princeton might be useful in this area.
I'm sure that when their turn comes around to teach, that they will not forget what they learn.
I'm sure David would say there's nothing that a student coming out of Princeton can't do. Let me just point out there are in this year there are 10 more of these lunch sessions, and I'm not going to permanently interrupt the questions, but I have to leave. And I did want to say before I go that over the next in the next 3 lunchtime sessions, we'll be hearing from Nathan Wolf, who heads up the Global Viral Forecasting Institute, talking about stalking. His title is "Stalking the Next Pandemic." In May, Dr. Leslie Vosshall from Rockefeller will talk about new approaches to preventing epidemics transmitted by mosquitoes. And also in May, Dr. Martin Chalfie, whom the name may sound familiar because he won the Nobel Prize a few months back, from Columbia, will be talking about molecules to spy on cells. Keep coming. Get your bosses here. Thank you, Dr. Botstein.
Oh, thank you. Thank you for coming. I appreciate it. Yes, sir.
So one of the factors with cancer is that they're not genetically homogeneous.
Yeah.
Right? And so I was just wondering, are there other cancers that, besides breast cancer, that may have maybe more challenging in that regard because their genetic diversity is greater and so may not yield the type of concise expression profiles that you can use in breast cancer?
That's a really interesting question. The answer is yes and no. No, in the sense that even breast cancer, we can still tell every woman's breast cancer from another cancer just by gene expression. There's a huge amount of individuality left. And so there's a higher order, subclasses, but at the detail, they're still all distinguishable. If you cut the tree, as we say, at about the same place, what you find is that most of the major cancers come in 2, 3, 4 or so subtypes of the very common ones. But there are some in which there's really only one subtype. So the answer is that is what I said, yes and no. The things that are really actionable, that really make a difference, the high-level differences, most of the anatomical tumor types, I would say, have 2, 3 different subtypes. And then a lot of long-tail outliers, right? Which, there's always, it's the way of the world. There's always long-tail outliers. Yeah. Yes, ma'am.
So if you could look in your crystal ball, what do you see on the horizon for some of the bigger technological breakthroughs that we'll be seeing within the next 2, 3, 5 years?
Well, I think the technological breakthrough that is just about is a tsunami of information of sequences that come out of the so-called next generation sequencers. This will take a little while. I mean, the water is building up behind the dam. The blockage is that no one knows how to analyze these data yet. And so, what's limiting actually is the bioinformatics and the statistics. I was at a meeting, and one of the big genome center guys showed a picture of his factory, okay, in which they had 1,000 sequencing machines, okay, and then he had an equal sign, and then he showed a blue box, which is about this big and this high. And they produce roughly the same amount of information. So there has been a huge change. And this blue box, although not cheap, cost $500,000, is very cheap compared to the $100 million investment in the factory, right, or more. Okay, so there's going to be a tsunami of detailed sequence information. And again, the issue there is going to be how to parse out that which you actually care about and makes a difference, and that which is just individual noise. And it's not straightforward how to do that. And there's a lot of discussion of which experiments make sense to do at this scale and which don't. but it is really it is as big a change as I've seen in my lifetime in magnitude. It's way beyond what anybody would have imagined. So I think that's clearly going to be one of those things. The other big thing I think you can begin to see is we don't have a really good theory for how complex inherited diseases are inherited. And the genome-wide association studies make it very clear that although you can find statistically significant things, they don't amount to much in accounting for the total variation in the population. And I think there are people out there who are thinking about this who are going to make some progress on that, I would guess, in the next 2 to 5 years. So I'm very, again, that's in the genomics world. I'm very excited about that. And then lastly, I think that the kind of field that is implicit in genomics, this sort of system-level integration, trying to understand how cells maintain their balance in changing environments, an area which my group is active in now. I think actually the tools are falling into place to make some progress there. And that, I think, will make a very big difference in the cancer business, too, because cancer cells are cells who've lost their balance in a way. So I think those are 3 areas that I can predict. Now, everyone should understand that that is the view from where I stand, okay, and what my field is, and that a chemist might give you a completely different answer and be just as right or wrong. Anybody else? It's 1 o'clock.
I wanted to thank Dr. Botstein. I also wanted to thank you all. The Congressional Biomedical Research Caucus, Congressman Holt, one of the co-chairs, for joining us. I hope to see you at future caucuses, and also thanks to the Coalition for the Life Sciences and Lynn Markey, who helps make these possible as well. Thank you.